to maintain quality. So instrumentation in food is important to bring about, good quality control. We also try to find out why it is important to prioritize quality control in food products. And we said because of increased regulatory action, people know a lot about food now, and people are more aware and want to demand the right thing. We also talked about consumer concerns about food composition and safety. We also talked about the fact that because now there's increased workloads in our various workplaces, more women are working, so people tend to buy food from outside a lot. And so it all boils down to health concerns. We continued with simple instruments. We talked about the measuring cup, spoons, weighing scales, and we talked about more complicated, equipment, instruments like the KIDA apparatus for protein determination, success apparatus for fat determination. We talked about some type of scales, trip balance, triple beam balance, torsion balance. We also talked about different forms of chromatography, the gas chromatography, paper chromatography, ion exchange chromatography. We talked about G-spec, gas chromatography, mass spectroscopy. We talked about all that, and I said, go and read more. Then we looked at food analysis. We talked about the criteria for selecting an appropriate technique to analyze your food. And we said breaking down the food components into the individual elements or separating anything into its parts is food analysis. We looked at how to evaluate products based on subjective or objective, how to record data, interpret laboratory data. We looked at proximate analysis, how to determine water or moisture content. We looked at determination of crude protein using KIDA methods, crude fat success method. We looked at digestible carbohydrate or crude carbohydrate through test of difference, meaning that you assess or you determine the amount of ash content, which is the total mineral content. You do moisture content, you do fat, crude protein, etc. And when you subtract from 100, the difference gives you that for the carbohydrates. We talked about different types of foods, the structure and chemical composition of carbohydrate, proteins, and lipids made up of oils and fats. We said these are all macronutrients. Then we talked about the micronutrients as vitamins and minerals. We talked about water. Then the study of the application of food enzymes. We talked a lot about different types of enzymes. Then we came to subjective objective evaluation of food, how to select the test type, and how to present your samples. For sensory evaluation, we said evaluation that is there a hand up? Yes, Anita. Anita, I see your hand up.
Madam,
Yes?
yeah, please, can you throw more light on the sensory evaluation of food?
Were you in class when we did it? Because today is not reviewing. Today, I'm just recapping so that we'll do what is left.
All right, thank you.
Were you in class when we did the sensory evaluation? You were not. Hmm. Okay, so we said sensory evaluation is a scientific method that uses the human senses to evoke, to measure, to analyze, and to interpret results using the human senses. Your eye, your nose, your tongue, your ear, texture, your touch. And these are usually based on how you You assess appearance using your eyes, aroma using your nose, taste your tongue texture by touching or breaking. And there are different methods. We have three main methods. Whether it's discrimination, where you need experts panel to tell you critical distinction between certain qualities of a product. And because you need distinct differences, you need people who are specialized, who know the product, who have eaten the product, are experts of the product. And for that, you use only 10 to 12. We also talked about the test of difference, where you just want to know how different it is from another product. And you need people who know the product, but not really experts. And so you can use 25 to 40. We also talked about the hedonic, where you want to test whether someone likes a product or not, and it's usually used for first new products when you introduce them. And because you do not need experts or people who know the products, numbers are always important. So you take a minimum of 75 and maximum of 150 to do the testing for you. We also talked about the presentation of your products. We said because these sensors of ours get tired easily, you do not provide your panel with too many products at a time. 3, a maximum 4, because when you give too many products, they tend to, get tired. Their senses get tired, and it becomes a problem. And so, you want to be careful. We also said that there are ways of presenting the product, either using the triangular tests where you have 3 samples, 2 are the same. One is a reference. We have the dual trial test where you present 3 samples. One is considered as just a reference, which is similar to one of the other 2. And you want them to select and make sure that they can distinguish between them. Then we have the 2 paired comparison, where there are only 2 products, and you try to, identify the differences between the 2 products. We also talked about some preparation, how you select your sample, how you train your panel, informing them of, what you mean by this color, that sound, texture, so that it will bring some uniformity in the scores. We said that you need to ask them not to use any perfumes, any perfume soaps, everything that will interfere with smell that day. You ask them not to, if they are on any medication, they should inform you, because certain medications like cough mixture, etc., can influence people's judgments, because there's this drowsiness, etc. When they are sick, they should let you know. During the testing, they are not supposed to communicate with each other so that they influence each other. You are supposed to provide a booth, water, code labeling so that they don't know exactly what the product is. You just find out from them if someone has any allergy for any of the ingredients, and you present it to them. You also have to add a questionnaire about how, it could be a hedonic scale. If you want to use all the 9, reduce it to 7, reduce it to 5, it has to be an odd number. And then they score for you and you analyze. Then you can find the implication of whatever they have done for you. And it's considered a scientific method. Then we also talked about the, what do you call it? We talked about the objective, where you use these instruments and equipments that we spoke about earlier. Are you okay with the explanation on the surgery evaluation? Anita?
Yes, madam.
Okay, thank you.
You're welcome.
Okay, and then we moved on to look at product development. I've talked about all this, the triangular test, the paired comparison, dual test. We said there are some disadvantages when it comes to what do you call it? There are some disadvantages in sensory evaluation. We talked about the subjectivity. Different people, have different feelings and opinions about all kinds of foods. Most of the time, it's very high cost, resource intensive during the recruitment, the training, and most of the time you need to compensate your panelists. Either you provide them with some snacks, some lime, something after they do it for you. The ideal time to have a sensory evaluation is early lunchtime between 11 and 12. You want your panel to come hungry. If they just finish lunch and they come, their senses will not be sharp to analyze your food for you. There's a potential of bias. Certainly, environmental factors could affect it. The temperature, the humidity, lightning, background noise. If you are assessing color and the place is dull, or you have some blue light on, something that can affect the color of your food material, then there will be bias. If the temperature is hot, if the environmental factors, you are testing ice cream, they bring it melted, it's not frozen, then you are not going to get the real ice cream that you need to assess. Sometimes sample preparation, there's inconsistency in sample preparation. The cooking times, the temperature, etc. Sometimes panelists fatigue, they are tired. It's time taking. You take one, you wash your mouth before you finish tasting, you wash your mouth again. It's quite tedious. And so these are some of the disadvantages of sensory evaluation. Before you conduct the sensory evaluation, you set clear objectives, you use controlled environments, you prepare your samples, you select your panel, and you provide clear instructions as to how they should evaluate the products. Then you analyze and interpret. Then we came to enzymes in food product development. We looked at natural catalysts. We said enzymes are natural catalysts. They improve the quality, the safety, and sustainability of food manufacturing. We said that these enzymes do help enhance flavor. They modify texture, increase shelf life, and sometimes are used to produce specialized products. We looked at baking, the common enzymes, natural enzyme amylases, which break down starch into simple sugars that yeast will feed on and cause fermentation and bloating, carbon dioxide. And so that your bread will rise. We looked at proteases, protein, enzymes, which modify gluten in dough. It increases elasticity, extensibility. It also brings about better consistency. We talked about xylinases, which increase bread volume. We talked about lipases, which act as emulsifiers. We talked about dairy chymosin, which coagulates milk during cheese making. We talked about lactase, which hydrolyzes lactose into glucose and galactose. We continue. We looked at lipases, fats, flavor development. We looked at tranglusaminases that improve texture of yogurt, etc. We looked at some beverages, some enzymes in beverages, the pectinases, jam, beverages, etc. The cellulases, hemicellulose, used in extraction of juices, etc. We looked at the amylases and glucanases used in washing, brewing, breaking down, pieces of meat, etc. And then we also talked about the advantages and innovation in enzyme technology. We looked at the fact of sustainability and efficiency, where these enzymes help us to cook or prepare food under lower temperatures and pH, reducing energy, water consumption, and also reduce the presence of bioproducts. We talked about waste reduction, because if these enzymes can extend the shelf life of products, then less will degrade and then waste will be reduced. We also talk about biotechnology and engineering, which helps to improve stability and activity. And then we got to research in food and nutrition. And that is where we got to the last time. And so today we are just going to talk about these things, research in food and nutrition, and we will be done. We are not going to waste too much time on this because I want to believe that you've done research methods. Yes or no? Have you done research methods?
Yes, madam.
Good. So
Madam.
this one, we are just going to talk about it, discuss quick, and we're done. When we started the course and we talked about experimental study of foods, we said experimentation, you control the variables, right? What are the different types of experiments that you can have? You are reviewing your research methods. So quick. What are the different types of experiments that we have? True experiments, quasi experiments. What are the differences? If you don't know, you check and we'll move on. Quick. What are quasi experiments? What are true experiments? I'm waiting for you. Yes. Yeah. Are we there? Are we there? Are we there?
Yes,
Hey,
yes,
who taught you the
Yes,
research
please.
method? So that I can go and tell the person. Mm-hmm. Who taught you the research methods? Mm-hmm. Mm-hmm. Okay, so when we talk about true experiments, they are scientific reason methodology or a method that establishes a cause and effect. In true experiments, because you are controlling certain variables, you want to see the effect of this on that, all things being equal, all this being stable or something. So it establishes a cause and effect, a relationship between variables. Okay, so usually we talk about independent variable. We have a control group, a random assignment group, etc. It's usually true experiments are usually used or considered as the most accurate form of experimental research. Why? Because it minimizes so many extraneous variables and you can be sure of the causality, cause and effect. So you either change or manipulate an independent variable so that you can observe the effect on the dependent variable. So, for example, there have been a lot of scientific research where they can give a placebo. A placebo is when they tell you that they are feeding you with something, but it is empty. There's nothing there. So they want to see whether the idea. I'll give you an example. One day, um, um, let's say someone went to the hospital, the theater for a surgery. And because the person was kept in the theater for 8 hours, when the surgeon, they brought her out and took her to the side ward. And she woke up from the anesthesia. The surgeon went there and said, "How do you feel?" She said, "I feel very well." Meanwhile, according to the setting, they couldn't even operate on whatever they wanted to do, because when they got there, it was a different story. But for the mere fact that the person thought, oh, they were going to operate, and when they operate, I'll feel better. Although the operation did not occur, the person felt, oh yeah, I am better. Hmm? Okay, so you want to see how um, an independent variable, what effect it will cause on a dependent variable. You have a control group. Control group is, for example, I want to test a cereal. Okay, I want to produce some baby food. In order that I can say that this baby food is good, apart from the nutritional content, I may have to look at the characteristics. Okay, because baby food has certain characteristics. It's smooth, it smells good, that's a good flavor, it's good taste, etc. I can use a well-known baby food like Cerelac or something as a control. So the people who are scoring my new products will be based on the characteristics of the Cerelac. So I use Cerelac as air control. Okay, so in true experiments, there are control groups too. Um, Sometimes we say that true experiments are more accurate because they are able to prove a definite cause and effect relationship. There's also high validity internally, um, especially when you use control groups. It helps to control any extraneous variables that may affect, and sometimes it helps to eliminate any unnecessary hypothesis, etc., etc., etc. So that is true experiments. And in the field of education, we do a lot of quasi experiments. What is a quasi experiment? Mm-hmm. Yes, research method. Oh, I wish I knew the person who taught you. Then I'll go and complain to that person. Mm-hmm. Yep. So what is a quasi experiment? It's a type of experimental design where you also have causal relationship. But usually in the quasi, it is between an intervention and an outcome. So in the quasi experiment, you don't do random assignments, variables. Rather, you look at intervention and outcome. There is no random assignments. So for you to conduct a quasi experiment, you need a pre-existing group that you can use. Some key features of a quasi experiment is the fact that your participants are not randomly assigned, unlike true experiments. So you don't have any random assignments. Instead, you have groups that are based on certain peculiar characteristics. So you can have schools, you can have, per a certain location, people from town, people from villages, people from this. And in the case of case experiments, to determine a cause and effect relationship, it becomes more challenging. Why? Because of probable differences between the groups you have selected. Sometimes for quasi experiments, manipulating independent variables is a problem. Okay. Most of the time, quasi experiments are used in education, public health, behavioral psychology, sociology. It has its own advantages and disadvantages. Advantages is that you can use it in a real world setting. They are more practical, less expensive than the true experiments. And because we said it's more of an intervention and outcome, it is ideal to study interventions in natural environments. The disadvantages is the fact that you cannot randomly assign. And so if there's no random assignment, then there's bias. Anything that you don't give people the equal chance, which is the random assignment, there could be bias. So sometimes research experiments get bias. Sometimes the results you obtain are more difficult to interpret. And the kind of statistical analysis are usually complex. And the conclusions are weaker compared to those of true experiments. Compared to those of true experiments. Okay. Okay. So we continue with our.
Yeah,
Mhm.
please, which part of the slides are you using? Because I can't trace it with the handout that I've made, the printout that I've made.
Yes, I am speaking. There's a slide 21 with experiments, with survey, with questionnaire, with interviews. Remember, I told you from the beginning that you cannot have everything on the slide. The slide is a guide. Did I say this or not? Yes.
Okay, thank you.
We are discussing. And that's why I'm giving you the opportunity to contribute. You have done research methods, and I'm asking research methods, and no one is minding me. Do you understand?
Thank you. Yes,
So it's
madam.
just like I'm speaking to.
Okay.
Yeah, it's that slide I'm speaking to. So I started with experiments. And I reminded you when we started talking about the course, and I said it's experimental study of foods. I asked you what an experiment is. Remember? We talked about the fact that it has to do with variables, dependent, independent variables. They have to be under control conditioning. It has to be under random assignments, etc. And I said there are two main types. You can have a true experiment, or you can have a quasi experiment. And I've tried to tell you the difference between the two, the advantages and the disadvantages. So that is the slide we are talking to. Exactly.
Yes, please. Thank you.
Thank you. Then we move on to the survey. Mhm. What are surveys? What are surveys? Mhm. What are surveys? Mhm. I wish I knew the person who taught you research methods, so I'll go and tell the person. I asked you questions and no one could tell me a word. Mhm. Yes.
Madam,
Mhm.
please, can we say that there is a method of research where we collect data by asking questions, a series
Okay.
of questions, yes,
Mhm.
from
Okay.
individuals, their
Okay.
opinions and then their behavior to
Mhm.
collect the data.
Okay. So in what way can you collect this information using questionnaire? So you could do even a face-to-face questionnaire. You could do an online questionnaire. You could conduct an interview. The different interviews could be by even phone interviews. You could have face-to-face interviews, and usually the survey, because they are itemized, they are items which are quantified. We say that you can use the survey research method to collect what? Quantitative data. quantitative data when you use the questionnaire, survey questionnaire. What about if you use an interview guide and conduct interviews? Then it becomes what? Qualitative. Then you can use that qualitative data only through what? Citing what some of your participants have said to back whatever conclusions you make. So one, if you want to collect quantitative data, then you use questionnaires where you ask a series of predetermined questions you have predetermined, and you have, some expectations in even the answers that will be provided. Then we talk about how to sample the group that you want to give the questionnaires to, etc. You could even, apart from interviews, you could use focus group discussions. The only problem with focus group discussions is you cannot assign the reason and discussions to one person, but rather a group. A group. Because when the question is put, several people speak and provide their opinion on whatever you are discussing. Okay. For your questionnaire, your questions have to be clear, concise, unbiased, and so that your participants or respondents can understand and give you the right feedback. If they don't understand the questions to put there, then you can imagine what they'll be telling you something different. Okay. We said that you could give a survey questionnaire, either paper, hard copy, or through the mail, through the telephone, panel surveys, focus group discussions, etc. Okay. Okay. Any more? Since you've talked about surveys in your research method, so you didn't talk about that. Okay. So you can have a questionnaire for your survey that uses a mixture of closed and open-ended questions. What are closed-ended questions and open-ended questions? When you say closed-ended question, what is it? Give me an example. What is an open-ended question? Give me an example too. Hey, are you Jew? Anyway, very soon you will learn because you'll be writing your exams. What are closed-ended questions and open-ended questions? Examples. Hey,
Madam, please, my hand is up.
Okay.
You have not called me.
Yeah, please talk.
Okay. I think an example can be, do you like eating
Tell me
fruit?
what it is first before you give me an example.
Okay, close-ended questions are questions that can be answered with a short or a fixed answer.
Mm-hmm.
So an example is, do you like eating fruit? So the answer could be yes or no. Or how are you?
Mm-hmm.
That's the close-ended.
Mm-hmm. And then the open-ended.
That one, it requires you to give more explanation to the question.
Okay, so more detailed descriptive response to your
Yes,
question.
yes,
Great.
madam.
Okay, that's great. Mm-hmm. Okay. Yes.
So,
So,
madam, do you want me to give an example of it?
well, you've given an example, a question that would demand yes or no. The person
No,
doesn't
for the
suggest no.
open-ended.
Okay, for the open, yes, you can go ahead.
So, an example can be, what do you like most about your school? With that one,
Mm-hmm.
you give more detailed explanation about what you like in your school.
Very good. I hope the others are following. Hmm. What about interviews? What are the different types of interviews that we have? Mm-hmm. Interviews. Mm-hmm. Mm-hmm.
Madam, please, can you repeat what you just said? Because my network was breaking.
We just talked about questionnaire, and we try to look
No, my network is not helping me.
at open-ended and closed-ended questions. Sorry. Hello.
Hello.
Yes. Yeah. So, well, I was responding when you said my network is not helping me, so I don't know what next.
I didn't get what you
But
just said because my line was breaking.
I'm saying that we haven't said much. We were talking about survey, and we looked at the further you can use questionnaire if you want quantitative data, or you can use interviews if you want qualitative data. And I said for the questionnaire, you could have close-ended questions and open-ended questions. And I read back to the class, what are close-ended questions and what are open-ended questions? An example. And one of your colleagues gave a good description of open-ended and close-ended questions and even gave us examples. So my friend, you may want to repeat your examples. Hopefully her network will not give her any problem so she can hear.
Oh, madam, it is me who gave you the explanation of the open-ended and the close-ended. But what you said after, I didn't get it.
I didn't say anything like that.
Okay, thank you.
So you see, you haven't lost anything.
Yes, please.
So the next question is, are there different types of interviews? Mm-hmm. Mm-hmm.
Yes, please. I think there are different types.
Okay, what are they?
I think we have physical interview, like the one you sit with the person one on one,
What are they?
then you interview the person.
Are they?
Hello.
Yes, what are they?
I said I think we have physical interview where you can sit with one with the person and then you interview the person. Then we have virtual interview. That one you do it online with uh, the one who is supposed to interview you.
Mm-hmm. Yes. Mm-hmm. What are the others saying? Is that what we have? Do we have physical and virtual? Have you heard of structured and semi-structured and unstructured interviews? Uh-huh. Have you heard of structured,
Yes,
semi-structured?
madam.
Those are the
Yes,
types of
madam.
interviews.
Okay, thank you.
Mhm. The mode in which you do it would be the face-to-face or the virtual online stuff. But the types are structured, semi-structured, and unstructured. So, what are structured interviews? Mhm. For every interview, you should have a guide, right? When you want to interview someone, you should know what kind of questions you want to ask. So, it's that structure, you know. So, usually, the level of flexibility or questions that you ask, which will, help you to collect whatever data you want, is either structured, where you use a rigid set of questions and you ask it in the same pattern to all your participants. When you do that, you'll be able to at least, what is it, compare. You'll be able to compare the responses that come along, isn't it? Because you're asking the same question. Ideally, when you have interviews, let's say interviews for admission, interviews for all those things, the best is to do that so that you have given all the people the equal opportunity to exhibit what they know. But if you ask different questions, it's not good. If you ask different questions, it's not good. You understand? Okay. So those are the structured ones. The semi-structured, um, you have some predetermined questions, but also you allow for some flexibility for follow-up questions. So someone will give a response and you say, oh, and you go further to ask something that will let the person explain it further. That's a semi-structured or semi-structured interviews. Okay. Then the unstructured interviews will be those that have no predetermined questions. And as and when it comes, into the person's mind, the person will ask you what we call the free flowing conversation. Have you heard of ethnographic studies before?
No,
Ethnography,
please.
ethnography. You've never heard it. It's a branch of it's a discipline on its own. You know, um, Historians and which other group of people, anthropologists, sociologists, they study culture and human whatever in detail. So I want to study how the Fulani live. I'll go there, live amongst them, observe how they eat, the kind of work they do when they take the head and cattle to the field and what they do. That is ethnographic, studies in detail. So you can use one person for an ethnographic study, where you study the person into detail. hmm Okay, okay. So under these three types, you could have even the focus group that we spoke about earlier, depending on how you present your questions to them, could be a semi-structured or structured or unstructured. You will have qualitative interviews where whatever information you demand is non-numerical. Okay. Any more? Mm-hmm. Any more? Okay. So you can look at the types of interviews based on whether it is structured, semi-structured, or unstructured. And you could also look at it in terms of having an in-person interview. But the in-person interview, it will depend on whether you are using set questions, which is structured, or you are using a mixture of set questions and allow the person to digress and talk more, or you are using the unstructured, the free flow, anything that comes to your mind you ask. Or you can have a panel interview where you have a lot of people talking to one person, and you can have even an informal interview. Okay. Hmm. Any more questions? Mm-hmm. Are there any more types of research instruments and tools that we've left out? Mm-hmm. 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 Any more? Mm-hmm. So, well, we say the methods go with the tools, isn't it? So, surveys, you could use either the instrument is the questionnaire or the interview guide, etc. Observation is another. You can use observation to collect data. From ethnographic, studies, a lot of observation is done. You systematically watch someone and you record the person's, way of doing things, behavior. Mhm. You could also do a document analysis where you are not collecting primary data, but already existing documents that you examine and extract things out to make um, some analysis and see what you call content analysis or document analysis or whatever it is. Mhm. Okay. Any more? So these are basic methods that we use. It's the same in food studies when you are studying food and nutrition. You can use any of these beyond the proper true experiments where you determine maybe the protein analysis or the protein content or fat content and all those things. You have what do you call it? that talks about some of these things. Any more questions? Yeah, any more questions? Is there something we have left out? Okay. I'm trying to reflect to see is there something I've left out in food and nutrition research? So these are simple things. Beyond, if you want to do more complicated research, then we are looking at what we call biomarkers. Biomarkers. What do you use biomarkers for when it comes to nutrition education? Mhm. Mhm. Biomarkers. That's a marker. Mhm. Mhm. Biomarkers in food and nutrition research. Mhm. Yes. So, but the biomarkers I'm talking about that we usually use in food and nutrition research um, are usually biological samples, right? So the blood, the urine can be used to tell the nutrients in your system. It can be used to tell your nutritional status. Remember, when we talk of nutritional status, we are looking at the person's, um, levels of all these nutrients, macro nutrients in the person's blood. Are they within the normal range? So you can use blood, you can use urine, etc. Mhm. So you can, for example, vitamin D, you can determine the level of vitamin D in your blood. You can determine iron levels, protein, potassium, etc. using urine. And several of these biomarkers, you can use them. Yes, anyway. Mhm. Okay, so I guess we wanted to look at I wanted to talk about experiments, survey, questionnaires, interviews. We've said so much in addition to what we've done in research methods. I think I'm confident and comfortable to say that we are done. If you have any questions, the platforms are there. We put the questions there. And as and when I'm a little free, I'll go to the page and respond to your questions. Any questions? If not, we would like to say a big thank you. Although I get very busy, you've tolerated me, and I'm happy that we've come to success. That is not to say when you have a question, you are not clear about something, you cannot ask. Feel free, put it on the platform, and it will do justice. Thank you. Hello, are you there? Are you there, please?
Yes, Prof.
Yes, Prof,
Yeah.
we are doing.
Okay. So if you happen to come up with a question, just put it on the platform.
Yes,
Any
Prof.
Okay,
clarification,
Prof.
any contribution, feel free. Once you put it on the platform, we all assess it and look at it. Thank you.
Thank you, Prof.
Bye bye.
Bye bye. Thank you.
Okay. Thank you.
Mm.